Okay, so now that we know what is an ion channel, now that we know that is what is a membrane potential, let's try to go back and try to understand what is an action potential. So if you remember, we were talking that action potential are rapid changes in charge across a membrane. So how you do that? Because you have different ions across the membrane, and the rapid changes are due to these ion channels that can open and it can move the ions across the membrane. Yeah, and these happen when a neuron is firing. So we're going to talk about four different stages. Maybe in some books you will find less stages, you will find more. We're going to focus on the book that for the anatomy and physiology course, and we're going to talk about first stage, that is depolarization. So you're going to break the normal polarization of the cell, yeah, and you're going to make it more positive. Yeah, so it was 7 minus 70, now it's becoming more positive. Then we're going to try to make it negative again. Yeah, and to some degree we go even further. Yeah, and we hyperpolarize. And then the last stage, the fourth stage, is the refractory period, that we are setting up everything to have everything ready to have another action potential if it's necessary. So let's start. So now let's talk about this all and none principle. That is very important to understand the action potentials. So imagine that you open an ion channel and there is only one ion that is going to move across the membrane and is going to produce a subtle change in the in the in the voltage in the membrane potential. So it's going to do something like that. What about, so would you consider that this is an action potential? What about if you do something like this? What about if it's a million? Yeah. How, what is the limit and how do you consider what is an action potential? An action potential, a nerve impulse, follow this non all or none principle. And in all this all and none principle, working to some degree similar to these long rows of domino that are standing. If you have enough energy to push the first one and the first one will touch the second one, it will create a cascade of events. Same thing happened with the action potential. If you have enough ions, yeah, to produce a change in the membrane potential, yeah, this will be enough to produce a cascade of events. Yeah, so I told you that the characteristic of a nerve impulse is known as an all or none principle. And is this all or none principle of an action potential is similar to push the first domino in this long row of standing dominoes. And when you push on the first domino, if it's strong enough, the second row is going to fall. Yeah, yeah. And so if you have enough um, changes in the membrane potential to produce the threshold of depolarization, it will be enough to move all the way. Yeah, if you reach this one, they will be enough to produce the action potential and trigger the repolarization, hyperpolarization, and the refractory period. Okay, now let's start talking about the depolarization. So to start talking about depolarization, the key thing that we are going to talk today is that this go from a negative to a positive, and how you do that, allowing the sodium to go inside of the neurons. Okay, and there are different ways in which we can do it. In your book, they mainly pay attention to the voltage gated channels. I'm going to show you some schematics that I have that I hope that they make it easier for you to understand. And they are also going to show the schematics from your textbook. So first thing that I want that you see is that I'm going to walk you through this diagram that I think it will be very useful, and because I'm going to use it to explain all the other phenomena. So here in blue you will see the outside of the neuron. So you will find this outer cell environment. Here you will find in yellow the inside of a neuron, or the cytosol. Here you can see 1, 2, and 3 ion channels. One is for sodium, that in the here and they tell you if it's open or closed. The green one will be of potassium, and the third one will be a sodium and potassium pump. Okay, so now the third thing that I'm going to tell you is that here they show you what happened before depolarization, and here they show you what happened after polarization. And here they show you the ions, yeah, some sodium, potassium, and how they are going to start moving. So the first thing that happened here is, as you see here very clearly, this channel of sodium is opening, and when it's going to open, there is now a path for the influx of sodium. Yeah, so if you see there is a lot of sodium here, there is a lot of sodium here. So what is going to happen? That the sodium is going to move to a place where there is a lower concentration of sodium. So it's going to move inside of the neuron. Okay, so before the sodium is out and the potassium is in, and we have a negative environment of -70 millivolts. Then, after the depolarization, as we open the channels, the sodium is going to go inside, so it will be an influx of sodium, and you will have an increase of 30 millivolts. Yeah, so it went from negative to positive. So look, here it was positive outside, and here the ions that are there will be negative. Now, what is going to happen is that we switch this. Yeah, so you can see clearly here is positive. Now the negative will be there. So what I told you in this slide is that depolarization referred to a sudden change in the membrane potentially, usually relatively, so you're comparing the inside to the outside, and we went from a negative, yeah, and later after the depolarization, it became positive, yeah, so there was an internal change. I also talk about that there was an influx of sodium, yeah, and this influx of sodium occurred because in the dendrites there was a signal that say open the sodium channel within the membrane of the axons, yeah, and you have all these processes taking place. Now I'm going to show you what happened, what they describe in the textbook. That is something very similar, but here in your textbook, again, you're going to focus on the voltage gated channels, and we're not going to talk about, for example, the sodium pump, the sodium potassium pump. So this is the schematic of your textbook, and now I'm going to walk you through again. So here again you have the extracellular fluid in blue, that in the other schematic was the outside environment, cellular environment. Here you have the plasma membrane, and here is the head, and here are the tails. So here is the head and tails of the phospholipids, and the cytosol here, that is the inside of the cell, that it will be located here. In addition to that, they show us two types of channels, one that is the sodium channel and the other one that is the potassium channel. The potassium channel only has two stages, one that is open and the other one that is closed. The sodium channel has two ways to regulate his activation and inactivation. You have one way to close it here, that is the activation gate, or one way to close it here, that is the inactivation gate. In this case, as the activation gate is there, this channel is closed. However, the inactivation gate is open, yeah, because it's not here attached here. So as one of them is closed, there is no flow of, there is no influx of sodium. So in other words, it will go here, the sodium, and they have to go back because there is no way to cross that area. So what I told you so far is that in the resting stage, your textbook focus on the voltage gated channels, and here they show you two of them, sodium and the potassium, and those both are closed, so there is no flow of ions. And in particular, they want to show you that there is two type of activation gates. Yeah, one, so two type of separate gates. One that is the activation gate, and the other one that is the inactivation gate. To open the channel, both have to be open. And in this particular case, I told you the inactivation gate, this thing, this circle here is not attached here, so it's not blocking, while the activation gate, that is structure that looked like a gate inside of the channel is there, so it's closed, so nothing can go through. And what happened now is that the polarization, we are going to open the channel, as I told you in the previous explanation. So look, it's exactly what I explained before. Now you can see in your textbook that the activation open, the activation gate is open. So look, now this move, and now the ion channel, the sodium channel goes there. The inactivation gate remain open, so both are open. So you can clearly see that the amount of negative uh, 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 charge is reducing, so the cell inside is becoming more positive. Okay, and again, your textbook for this diagram is going to try to simplify things. So, for example, it's not going to talk about these sodium and pump ions, although they said that when you open these sodium pumps, there will be these gates, voltage gates, they could allow to get so much iron that it could be very bad. However, they also said that if that and these sodium pumps are working there, so you only were going to see a sudden change, that is the subtle change compared to what could actually be happening if you open all those channels like that. So I told you that in the activated state of the voltage gated channel, both the activated and the inactivation gates are open. So there is an inflow of sodium. And I also told you that these sodium pumps, sodium and potassium pumps, are very important. And if we let all the sodium voltage gauge open, there will be just too much sodium. Yeah. So what is happening is that you have these sodium and potassium pumps also working. So there is not a really bad increase in sodium. Yeah. So it allowed to maintain a low concentration of sodium inside of the cell. Okay, so now I explain what happened with the depolarization, this rapid change in sodium, yeah, and I explained it with these schematics, and I also explained it with the schematics focus on the voltage gate, that is what your textbook showed. Now let's move into the next, that is the process of repolarization. So let's talk about the process of repolarization. So in this section, the main idea is we know that cells were depolarized, so now we change the concentration of sodium inside of the cell, so we increase the positive charge. Now we're to do the opposite. We're going to make it more negative, and how we do that? Not by removing sodium, but by removing the potassium. Yeah, so it will become more negative because we're removing the potassium from inside of the cell. So again, this is the same diagram, and I'm going to keep showing you this one again and again. So here, uh, before the repolarization, yeah, you have a little bit of potassium. You have here in this diagram 1, 2, 3 potassium here. So when you open the channel, what you start with a positive charge with a 30 millivolts, and now all the potassium is going to leave. So instead of 3, you have 1, 2, 3, 4, 5, 6, and 7 potassium outside. Yeah, well, inside you will only have only one. And again, this is just a diagram, but the idea is very clear. After the process of repolarization, you have a potassium efflux. Yeah, so if you remember, it was negative before, and here it was positive here inside, and we're going to do is try to repolarize, so to give it the right polarity. So now this is going to become positive and this is going to become negative, and this is exactly what happened after the repolarization. Now you have so much sodium, so now outside of the membrane you have charge that it will be positive, yeah, and here you will have charge close to the membrane that it will be negative. In addition to that, remember, sodium is already closed. The sodium pump, uh, sodium potassium pump is also closed. The only thing that is open is the potassium channels. So let's summarize all the things that I told you. I told you that the process of repolarization, it refers to the restoration of a membrane potential following the depolarization. So I show you that we are restoring a negative internal charge. And so we went from being positive before in the beginning of repolarization to come back to be negative in charge after the repolarization. And of course, we're talking about the internal charge, the one that is in the yellow area. And I told you that all this, we did it by opening these potassium channels, and there was a passive efflux of sodium. Why? Because there was very little sodium before here, and now, so there was diffusion here. Now you have a lot of sodium, so it's a passive efflux. And as a result of that, now the internal environment of the neuron become more negative again, and this is how you repolarize the membrane potential. Now I'm going to explain the same thing, but using the voltage gated channels that are explained in your textbook, but it's the same idea. So the textbook wanted to show you three things here. First thing, I want that you see that the activation gate in the sodium is still open. Yeah, still open. However, the inactivation gate now is attached here, so there is no more sodium. Yeah, it will have to leave because this one is closed. The other thing that is showing you is that now you have the potassium channels open, and now they can leave. And they will go outside because there is, as I said before, there was an efflux of potassium because just diffusion. So as a result of that, if you go to this diagram, you see that they are start becoming less positive, and you can even see in the graph that some negative charges start going closer to the membrane because they are free. So here you have some positive and some negative, but later you will see that it will start becoming gradually and gradually more negative. Okay, so let's let me show you where your textbook wanted to that you remember. So first thing that it told you is that the repolarizing the phase begin uh, by closing the inactivation gate, and I show you that. Second thing that they show you is that the potassium channels are open and that the potassium channels are leaving the neuron. And here you see that the negative charges start locating or building up along the inside surface of the membrane. Now let's see what happened at a later stage of this repolarization. So here we have the later stage of repolarization, and here what happened is that as the phases continue, you see first here they're showing you this is still open, so there is still an outflow of potassium, yeah, and the more that it leaves, the more negative the charge becomes. So here you see here that it will continue going down, and here you see that the charges in the membrane are still negative here, and now all of them will be positive here. So it is still going, it still is resetting the system, and it's not only resetting that, it's also resetting the sodium channel. So look, here is still closed, yeah, so they cannot go through. However, now the activation gate is the one that is closed, yeah. So now this activation gate is the one that is closed, and the inactivation gate is open. Okay, so again, is start setting up everything to for the next action potential. So we already talked about the process of depolarization, that it was the increase of sodium inside of the cell that led to an increase of a positive charge. Then we talk about now we are going to repolarize it to come back to the normal polarity. So how we do that? We are making it more negative as it shows here, and we do it by opening the channels of potassium. So we are reducing the amount of potassium inside of the cell and increasing it outside. Now we're going to talk about the process of hyperpolarization. If you see the process, the cell become even more negative before starting again and resetting the entire process. So let's talk about that. So in your textbook, they don't show a lot of the process of hyperpolarization with diagrams, but I'm going to show you what happened. So what happened at that point is that, look, in the beginning, you still have opened this channel. So what happened? You are losing still a lot of potassium, yeah, and it goes beyond the minus 70, yeah, it can go even more negative at about minus 90. So what you do, you need to close it here, and remember this channel doesn't go in, it's not like the sodium channel that have an activation gate and an inactivation gate. This one, it has open and close, so it's a binary channel. um, so as this channel close, we're going to go through other process that I will show you in a second that allow the cells to reestablish the normal membrane potential of minus 70. And remember, as I said before, your textbook wanted to remind you that the voltage gated channels, they alternate between closed in the resting stage or open in the activated stages. Okay, so now in the following and last stage in the refractory period, I just wanted to show you how the cell is able to reestablish the distribution of sodium and potassium outside and inside of the cell. Okay, so we are done. We already know that how a stimulus take place and generate a nerve impulse by depolarizing and then repolarizing the cell. Then it goes to a hyperpolarization, and finally we're going to talk about this last stage, that is the refractory period, where the cell completely reset the membrane potential. What is important here? Um, I think the most important part here is that you need to remember that until this cell is completely reset, the neuron is completely reset, it will be more difficult to initiate a nerve impulse. Yeah, so there will be stages in this. For example, around here will be very difficult to initiate a new nerve impulse. The closer that you are to this normal stage, the more likely is that another uh, nerve impulse take place. It will just require a larger stimulus. This refractory period, uh, depending on the textbook, there is a little bit of variation. I'm going to show you again some general information, and then I'm going to show you what appear in your textbook. Now let's talk about what happened at the cellular level during the refractory period. And I remind you, the refractory period is the time that follow the nerve impulse, and the neurons need to set up everything again to be able to fire a new nerve impulse. Okay, and how we do that? So we can see that the charges outside and inside are correct, and after that they remain in the right way. So even before the refractory period, the charges across in both sides of the membrane are correct. The other thing that we need to change, it was maybe the inside of the membrane. It was negative and it was very close to -80, but it needs a little bit of fine tune that occurred during the refractory period. Okay, so it seems that there are just minor changes. So what is the major change that happened? So the major change that happened is that here, if you see, there is a lot of potassium. We needed to bring it inside again against the gradient, and the sodium that is inside it needs to go outside. Yeah, and this is the normal distribution that we had in the beginning. Okay, so to do that, we don't need the sodium or the potassium channels that we talked. We need, as we go against the gradient, we need this. We need the sodium and potassium pump that require ATP. Why? Because as we are we cannot move things passively anymore. We are moving against a gradient, and for that we need ATP. So, in other words, we need to take the sodium out and put the potassium in, and this is the major changes that happen. Okay, so to summarize, I told you that this period is very important because it allowed the neurons to set up everything to fire a new nerve impulse. And what is the main thing that happened? Yeah, we need to reset the the ions distribution. So the sodium has to go outside and the potassium ions need to go inside. Yeah, and so if you remember the first slide that I showed you, this will be exactly like this. Okay, this will be the ion distribution, and this will be the -70 millivolts. It was exactly the voltage that I told you. And if you remember, in the beginning during depolarization, we have sodium influx, and then during repolarization, we have potassium efflux. So right now what we have to do is to fix this ionic distribution because it was reversed. Although we had the correct charges, we definitely needed to bring those ions to the correct distribution, and to that we need this sodium and potassium pump that it will restore the resting potential to the initial conditions. Now to finish, I wanted to show you this diagram again, but color coded. And again, this is taken from your textbook, so we're going to focus on the voltage gated channels.
So let's start. So in the beginning, you have this purple area, and we're going to go back into this purple area, yeah, light purple. So what happened here is that we are in a resting stage, and we are before the stimuli. So what happened? The resting membrane potential is there. The channels are in a resting stage, or the channels are closed. Then you go to the blue area and the green area, and it's very clear what is happening here. You have a small channel change, and when it goes beyond the threshold, you go for a quick change, and you have all the sodium going inside of the cell. And this is what your textbook tells you. A stimulus appear, and if you go beyond this threshold, you trigger that the channels, the sodium channels open, in particular the activation gates open. Later in the two orange areas, you start the repolarization, you make the cell much more negative again, and you do it in the beginning, in the early stage, by removing the potassium, by opening the potassium channel and closing the sodium channel. And these two stages, the light green and the light orange, are called absolute refractory periods. Why? Because there is no way that in this stage, yeah, you can have a new nerve impulse happening. Yeah, at this stage, the cells are not able to do that, and there seems to be a correlation. So when you go and look at the voltage gated channels, you find that these ones, the inactivated gates are closed. Yeah, so and they cannot reopen in that place. So you cannot initiate a second nerve pulse. Yeah, and it seems that is because these inactivated gates cannot open. The last stage, this dark orange, here it has the late repolarization, the hyperpolarization, the refractory period, and you're going to go back into the resting stage. In this stage, if you only focus on the voltage gated channels, they seems to be still open, but and the sodium channels are are already set up in the resting stage. And in this case, yeah, you have, they are called relative refractory period. And in this case, that period. Now to finish, here in this section in purple is called a relative refractory period, and as I said, you can initiate a second impulse. However, it needs to be larger than normal, so it has to be a very strong stimuli. And they want to remind you that although the sodium channels are already in the in the resting stage, the voltage gated channel, remember here we're not talking about the sodium and the potassium channel, we're only talking about the voltage gated channels, they can be still open. Okay, and with this we finish this section, and if you have any questions, let me know. To finish this section, I just wanted to put all the information that we have learned in this class and try to see it in a neuron, because I was thinking that, for example, all the process happen in different neurons. So one neuron is depolarized, the other one is repolarized, the other one is hyperpolarized. What actually happens is that axons of neurons can have different length. So some of them could be few micrometers, some of them could be even centimeters of length. So you can have in a single axon different process happening at the same time. So if you remember, the dendrites start transmitting the signal, and all of them zoom and combine here, and now they're going to move in through the axon. Yeah, but they don't move in a single stage. The axon can be can have different stages at the same time. So for example, here in the beginning, what you're going to see is that although the action potential is traveling in that direction, this section is already positive, yeah, and it's already depolarized. While this region is in a resting state, sorry, and the membrane is already is negative, yeah. The inside of the neuron, the charge will be negative. Now let's see what happened later. So here we have time 1 that I already explained. Here we have also time 2. And if you remember, I told you that you can have one stage and another stage here. Yeah, so there could be a combination stage in the same neuron. Here you have not only 1, 2, but 3 different stages happening simultaneously in the same neuron. So if you see here, the depolarization is spread, and now this one is that it was in a resting stage, now is depolarized. And the third region, there is still some areas of the axon that are still in the resting stage. And the first region, the blue region, here is not depolarized anymore. Yeah, here the channels have closed. Yeah, and now we are trying to come back into a negative charge inside of the cell. So remember, I told you that there could be multiple things happening in one single axon, and that the depolarization is spread down the axon, and that the first part where there was depolarization before, now you have is repolarization, and this happened because the sodium channels are already closed, so the cell, the neuron cannot depolarize again. Now it needs to move forward and start going to the process of hyperpolarization and refractory period. So here, in addition to time 1 and time 2, here you have a time 3, and again, same thing, multiple things can be happening at the same time in one axon. So now, look, you remember the one that close to the soma was depolarized, now it's in a different stage, now it's in a resting stage. The one that it was depolarized before, time 2, now is repolarizing, and the one that it was in a resting stage now is moving to depolarize. Okay, so it's pretty straightforward what is happening, and depending on how long the axon it is, you can have even more stages, but this is the main idea. Okay, I hope that you enjoyed this. I know that there is a lot of information, so please give a look to your textbook, and if you have any questions, just let me know. Have a good day and bye.